Short tandem repeat polymorphism. Short tandem repeat polymorphism, they are also called as simple repeats. They can be in the form of mononucleotides, they can be in the form of dinucleotides. Mononucleotide is just the repetition of a single nucleotide. Like if we take the example of adenine, so adenine will be repeating each time A. So dinucleotides, the meaning of the dinucleotide is that two nucleotides, they will start to repeat on the DNA sequences. Like if we take the example of AT. So AT, there are two nucleotides, there will be repetition of AT and so on. So these kind of the sequences, they are called as dinucleotides. While in case of trinucleotide, there will be repetition of 3 nucleotides. Tetra, there will be repetition of 4 nucleotides. So these kind of the sequences, they are called as simple repeats and they are called as STR markers. So different alleles, they contain different number of the repeats. Some individual, if he has 2 allele, 1 allele of the father and 1 allele of the mother, it can be that on 1 allele there would be 7 repeats, while on the other allele there will be 9 repeats. If on 1 allele there are 7 and on the other allele there are 9, we will say that the individual at that particular locus is heterozygous. While in case if both the repeats on the DNA of the mother and DNA of the father, the individual has obtained both the DNA, if the repeats they are same, that will be called as homozygous. Here we can see to understand the phenomena of STR polymorphism. Here we can see that there are 2 DNA, DNA 1 that is of individual 1, DNA 2 that is DNA of individual 2. In this case we can see that there is 4 nucleotides, that is tetranucleotide. So there is a repetition of tetranucleotide. Each block represent 2 tetranucleotide 1, 2, 3, 4, 5, 6 and 7. So there are 7 time repeat of AATG. So 7 repeats of A, T and G in individual number 1. While in case of individual 2, there are 8 repeats, 1, 2, 3, 4, 5, 6, 7 and 8. So in case of individual 2, there are 8 repeats. So these are heterozygous because on 1 allele there are 7 repeats, while on the other allele there are 8 repeats. So if both alleles are of same length, then they will be called as homozygote, while if they differ from each other, then they will be called as heterozygote. So in this case we can see that individual 1 has 7 repeats, while individual 2 has 8 repeats. So these individuals, they are heterozygote. STR markers are used in the DNA forensics, particularly for the criminal justice, civil justice, as well as for the paternity analysis. So these are some of the common STR markers which are used in the DNA forensics, and we can see that these STR markers are present on almost all of the chromosomes. We can see they are present on chromosome 2, 3, 4, 5, then 7, 8, present on chromosome 11, 12, chromosome 13, 15, 16, 18, 19, 21. And if there is sex typing to identify that the individual is male or female, then these STR markers, they are used for the sex typing. So STR markers are short tandem repeat sequences which are used in DNA forensics. All these markers which are used in the DNA frequencies, they have multiple alleles. So in case of all these markers, we can see that have a lot of variation. In this, let's say in this marker, we can see that there are 11 alleles of this marker. So there are 11 different fragments or the repeated regions which are present in case of this marker. So all these markers, they are highly polymorphic. We say highly polymorphic, and highly polymorphic markers are used in case of DNA forensics. Here to understand short tandem repeat sequence, that how there are tandem repeats. In this case we can see that there is a repetition of AGCT, again AGCT, again AGCT, again AGCT. So there are 4 tandem repeats of tetranucleotide. Because these are 4 nucleotides, we will say that it is tetranucleotide. While on the second allele of that individual, there are 3 tetranucleotide, AGCT, AGCT and AGCT. So here there are 4 repeated regions of tetranucleotide. Here there are 3 repeated regions. The tetranucleotide has been repeated twice in case of allele 2. So there will be the variation in allele 1 and allele 2 based on the number of repeats which are present in allele 1 and allele 2. How these alleles, they are separated from each other? If a genotype has 7 and 9, let's say this is for the repeat number 5, this is 6, this is 7. So one allele is present approximately at band number 7 and the second allele is present around about to 9. So genotype will be marked as 7 and 9. While in case of second locus, we can see that one band is here, that is in the approximate direction of the 6. So it will be 6 and this one will be 8. So genotype will be 6 and 8. In this case, allele 1, the genotype will be 7 and 9. While in case of allele 2, the genotype will be 6 and 8. In this way, these fragments, they are amplified by the polymerase chain reaction and run, and once they are run on the gel electrophoresis or on the DNA sequencer, the genotyping is performed based on the repeated sequences. So short tandem repeat sequences are the simple repeats. They are usually in the form of mononucleotide, dinucleotide, trinucleotide, tetranucleotide.